So I'm here with Chris Ponting from the University of Edinburgh. We're discussing the DecodeME study. The preprint was released this week, and so caused a lot of stir. So Chris, thanks for talking with me. Why don't we just get right into it and tell us, the groundwork for the study, how the study came about, and then what the goal was of the study and why it was important.
Thank you for the invitation to speak to you today. So the, the, the study really came from a researcher patient charity collaborative that Stephen Holgate set up with Sonia Chaudhry. It's about 10 years ago. And it stemmed from that and the conversations that came from that and um, engagement with the Medical Research Council and the NIHR, who ultimately funded this, in an iterative fashion that meant that we wrote several documents, answered lots of questions, talked to many people, and ultimately were invited to apply for a strategic award, which we did and did so successfully for a genetic study. And why genetics? Because from my point of view, I had not seen a persuasive argument as to why ME is destroying so many people's lives. And I'm sure people will disagree with me, but there's a rather scattergun approach to ME research. And so what I think still think that the field needs is a more evidence-based approach to determining what are the right experiments to do first. And the best way of doing this is a genome-wide association study, and most diseases have benefited hugely from these GWAS 10, 15 years ago, and ME had not.
So is it basically that genome-wide association study? I mean, would every disease would have some genetic predisposition likely, or any condition would likely show up with some sort of findings on a study like this?
So if you have sufficient numbers, you will, I believe, be able to find genetic predispositions or susceptibilities to diseases and changes in any trait in the human population. So yes, there are genetic predispositions, and we wanted to find out what they are for ME.
Now, is that the same thing as saying those are causes? How would you sort of parse that sort of relationship?
So I have argued that the associations that we have found represent causal associations.
Mhm.
And the reason I'm making that argument is that possible confounding factors, in particular differences in genetic ancestry between our cases and controls, have been dealt with
Okay.
in the way that we analyze the data. So moving those confounders to one side, I believe that the associations are indeed causal.
And does that mean that we'll go on to discuss what they are, but does that mean just in principle that one has to have these genetic variations, or it just makes it more likely? Like if someone's sick, does that automatically mean they would have these variations, or does it
Good
mean
point.
they're more likely to have them?
Yeah, it is indeed. It's about uh, changes of risk, and discussing risk is quite difficult to understand. So there's a slight tilting of risk if you have one genetic variant over another that might change your odds ratio. You know, the relative risk that you will have this diagnosis by even a percentage point or
Mhm.
two. So it's definitely not definitive that if you have a variant, therefore you will have ME or you have ME.
Or
It's
if you don't,
definitely
if you
not
have
diagnostic.
any, or if you have any, you don't necessarily have every one of these things. It's a more population level finding.
And therefore it's really quite hard to convey not just what we're doing, but why we're doing it. Because if it's not relevant to individuals, as we're saying, then a lot of people quite rightly ask the question, well, why are you doing it then? And the answer is we're doing it because it's relevant to the population. And we're trying to understand what are the genetic factors that, on average, contribute to people's risk of ME.
And by
Um,
and then you
and
can.
therefore, that's where we can start in the future to develop new programs of experimental science.
So basically, the idea being that these genetic markers, whatever they're coding for, the things they're coding for are somehow influencing the course of the disease. And if you can figure out what they're coding for and what exactly is going on
Yeah,
those genetic signals or loci, as you call them, that you can then figure out something about the processes that are causing the
yeah,
symptoms and
yeah.
causing the disease.
And absolutely. And I'd like to add one more thing, which is the effect sizes are tiny, but if these effect sizes tell you where to intervene, then sometimes using drugs you can intervene to such an extent that the effects of the drug are enormous.
Okay.
So just because the human genetics gives us small effect size does not predict the effect size of an intervention like a drug.
Okay, so that's sort of good background on what this means. And so tell us what findings from a study like this don't mean, and then we'll go into the actual findings. Like what
Mhm.
can't say from a study like this? And I think some of the criticism, at least from the one person at the Science Media Center, was, well, we don't know what it means, and it may mean nothing, and it's very small anyway, and these things haven't, amounted to anything. So what can't we tell from this?
We can't tell whether any individual person has ME or will come down with ME or not. We can't tell that. We can't tell what will be the effective drugs that will be developed in the future. I very much hope for ME. It's a starting point. It's not the middle of the story. It's not the end of the story. It's the beginning.
So I wrote something like it provides clues, it doesn't provide answers. Is that sort of a sort of way of describing it?
The analogy we're using is that we're providing the X's that mark the spots on a treasure map. And what is now required is a bunch of scientists to go and to dig and to find what the treasure is and to bust open those treasure chests.
Okay, so to do the study, you had a national, or I mean, I guess a UK-wide effort to get saliva samples. And so tell us the final numbers you came up with and then what the findings actually were when you did your scrunching and analysis.
Yeah, so we recruited over 25,000 people to the study, which is fantastic. And that was due to the co-production with people with ME and ME charities, gaining the trust of so many people that they would wish to contribute. Around 21,000 were eligible for the DNA portion of the study, and we received just over 18,000 people's DNA samples through saliva kits. Then it went through a lot of quality control, both quality on the samples, quality control on the data. And for this initial analysis, we've restricted ourselves to Europeans only by ancestry. And you'll remember that what we said earlier about causality, we have to be very careful that we match our cases and controls so that we can then make these claims about causality. So that's why we've kept to the majority population of people of European ancestry.
Mhm.
Okay, so then we have the around about 500,000 places in our genomes where we have people's DNA letters and their differences, and we compare them against the general population from data that's been collected over 10 years ago by the UK Biobank. So we didn't need to pay large amounts of money for that. And we compare them to basically ask the question, is a genetic difference more often seen among people with ME against population controls? And because we're doing so many tests, 500,000 or so, we have to have a very strict statistical threshold
Right.
to say that something is significant. And
Because
that
you have so many tests that so many things could come up just by random.
Yeah, exactly. If you do 100 tests and you employ a 5% threshold, five of them will come up as significant just by chance. So there's an industry-wide significance threshold that's been established for many years, which is 5 times 10 to the minus 8. So we use that, and after applying that statistical threshold and controlling for most things, we came up with 8 places in the human genome where there are statistical associations to ME risk.
And these are collections of genes, not a specific or a sort of an area as opposed to a specific gene.
That's right. And these are areas that lie between genes, that lie across many genes, that intersect genes. So what we can't say is that we have an association to a gene.
I see. Okay. And so these 8 genetic signals or genetic markers or whatever fell related to immune function, as I understand it, after infection, as well as to nervous system function.
Yeah. So if we ask the question, where do the genes that lie in these signals, where are they more often active in the body? And we do that using an objective statistical test. The answer is very clearly in the brain and across the brain, 13 different places. So we have strong evidence, therefore, that these associations relate to neurological processes.
Mm-hmm.
That's where we start. And then when we look at what the genes are, there are several that are provide particularly good explanations of what ME processes could be going on. For example, there's one near a gene called OLFM4, which its protein is involved in suppressing the immune system. And that, therefore, if it goes wrong, could lead to all sorts of problems with the immune system. And that is one of several such examples that could be true, but don't have to be true. So let me see. There's another one on chromosome 6, which is right next to a gene called BTN2A2, and it works with similar molecules for sensing bacteria, and also has a role in T cell receptors. So again, immunological explanations. And we can go through and look at around 50 or so genes and make claims as to whether these are the genes that are causal.
I mean, that are around these areas.
Yeah.
Okay.
So there are about 50 that we have written up in a document that goes with the preprint, trying to summarize what the evidence is. And in future work, we're going to hone that to try and do better, to restrict our numbers to fewer genes.
So what's your sense? I mean, it sounds like from your perspective that the sites that you found correspond to the kinds of complaints that people are having, the kinds of symptomology that people have. Is that fair to say?
I think it is. It was the penny dropped with me in particular that what we were seeing was that the DNA was speaking back to us what we had already been hearing from people with ME over their lived experience. There is another genetic signal which is relevant here, CA10, where the genetic signal overlays very precisely a previously observed genetic signal, not for ME, but for chronic pain.
Okay.
And so exactly what we're seeing here is the genome variation speaking back to us about chronic pain that we have heard about so often from people with ME.
Mm-hmm. So, what's your sense of the reception this week?
We have been overwhelmed by the reception from people with ME.
Well, that I can imagine. That obviously.
Absolutely. Okay, so let me just state that very clearly that we have been overwhelmed because that really means a lot to us. Then moving beyond that to.
You had any concern about sort of the overinterpretation or excess hopes? You know, people thinking, oh, next week there's going to be a treatment, or people overinterpreting perhaps what the current significance of the findings?
I haven't seen very much of that. I'm always impressed by people with ME's understanding of the current state of the art in science. Science is complicated. Outside of my own area, it's difficult for me to understand, and yet so many people have got on top of the issues, the science, their pros, their cons, their limitations, and have been communicating within the community to come up with very similar statements to what I believe. It's been lovely to see.
So that's the patient reaction. You've been overwhelmed by that, by the
Yeah.
community and so on. How about professional side?
Um, Wonderful, to be honest. So we had sent the preprint to independent researchers ahead of releasing it because we didn't want to have made a major mistake. And we had really good reception among our independent researchers when they came back with their comments, which we then changed the manuscript in response. And then our international colleagues who are authors, fantastic response from them. And then there are two out of three, I think, on the Science Media Centre who are very supportive of what we were saying. And what they were saying with respect to the limitations were fair.
Well, no, I think that I mean, obviously, limitations are essential, and it's important to make clear what the study can say and what it can't say. Um, I think the third comment there was basically all about what it couldn't say or what it probably wasn't saying or something, and it didn't seem very thoughtful. But as you say, the other two from the Science Media Center, I did think were quite thoughtful responses. Um, what there's been a lot of media coverage too, at least in the UK. I haven't seen much in the US at this point. I don't know if there has been. Um,
Newsweek magazine and Science magazine have covered it.
oh, Science did. Okay, great. Yeah, so those are more sort of international. Okay, so what happens next?
So we're what we've written down and what we've shown, we believe in, absolutely. So what our future work is all about is adding to that and improving on what we already have done. There are some studies that we haven't done, and we are quite open about that in the preprint. We haven't yet studied the sex chromosome, the X chromosome. We haven't done that. And we haven't completed the particular analysis on a region on chromosome 6 called HLA, which has given us a result, but one that is confusing, and therefore we need to look into it much further. We want to, and I said this earlier, want to improve our understanding of what genes could be involved in ME risk. And we're going to also compare the genetic signals in ME with other traits and diseases.
So what's the status of the journey to publication of this paper? Has it's been
Yeah.
submitted or it hasn't been submitted yet?
No, 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 it hasn't been. We will only submit it when it's ready for peer review. And DecodeME hasn't even talked about how we're going to do that and when we're going to do that.
So this form, it won't be submitted in quite this form. It's still going to go through more revisions before you submit.
Yes, absolutely. And just to be clear, the reason why we've done it this way, the major reason is that we always said that it wouldn't be peer reviewers who would read the results first. It would be people with ME, including the participants of DecodeME. And we're also coming to the end of our funded period. So we need to produce something by the end of that funding period.
So do you have an anticipation for when, I mean, are findings like this sort of actionable by anybody until they're actually published? I assume people have to wait till there are other people who want to sort of start doing research. How does that work?
Well, no, I mean, it's out there. We're providing all of our data, all of our findings online so that people can then action them. They can take them forward if they are convinced by our science
Mhm.
and they wanted to make good use of them, then brilliant. I'll be really happy to see that happen. If they are not understanding of our science or disbelieve it and they want to wait for peer review, that's up to them too.
Do you have any predictions for how long that process will take?
Um, Unfortunately, some journals can take over a year
Right.
for dealing with a manuscript. We had one that was published earlier on this year that lay unreviewed for 12 months.
Oh, wow. Okay. That's a problem.
Yeah.
Um, so finally, what does this do to the argument that MECFS is a functional disorder or, a functional disorder, whatever the term functional is currently meaning? What does this have to do with that idea, or what does this say about that construct, that there's not really um, anything pathophysiological one might find, and that there's other things going on that are causing symptoms?
Yeah. Well, every trait, every disease will have genetic contributions to them. So just because ME has genetic signals doesn't make ME really any different from any other disease or trait. So what we are doing is asking the question whether the associations we are seeing are equivalent to what others have seen for other diseases. And thus far, the ME ones seem to be specific to ME and not to any other trait or disease, aside from this one of chronic pain.
Mhm. And that means what in practical
Well,
terms?
it means that the ME genetic signals don't are not equivalent to any for, I don't know, arthritis, Parkinson's, Alzheimer's, depression, anxiety, none of those,
Mhm.
and more.
Okay. So you've been doing this now for what year did it start? And now you've sort of
5 years.
disgorged this mammoth thing. How are you feeling?
Exhausted, but relieved that there were genetic signals.
When did you
That
know
was
that you had them? When did you know that you had found? When did you find out what the findings were? How long ago?
science is often portrayed as having eureka moments. It's never like that, to be honest. You edge forward, you make claims which not all are then found to be true. You then substantiate the ones that you think are true, and you get more and more evidence until ultimately you're convinced. But it's incremental rather than being a step change. We had the first piece of evidence that we were seeing true genetics around about September, October last year.
Okay. Now, how does this get replicated in other cohorts? Is that also, obviously a process that has to happen? I mean,
Yeah.
you looked at some other biobank material. Um,
And I missed it out on the things that we need to do. We already knew that what we need to do is to compare against UK Biobank and cases who have been defined in a particular way that we're most interested in. And unfortunately, for technical reasons, to do with the fact that we're using whole genome sequence and not genotype data, that has proved to take far longer than we suspected. So that is another thing that we need to do before sending to a journal. However, in the preprint, we have colleagues from around the world who have essentially done lookups that they have already done a genome-wide association study on various things, and they've gone looking to see whether the signals that we see, they also see in their data. And among the eight that we highlight, four of them are significant, but only at the nominal level of less than 5%. In other words, this multiple testing correction thing we talked about
right.
earlier, they don't survive that correction. So there's an enrichment of small p-values for the statisticians among us, which is meaningful, but it doesn't say which of the signals that we have reported have been replicated.
I see. Okay. Um, that's a little complicated, and some will understand that and some won't. Anything else I haven't asked that you want to make sure people are aware of?
We have found no evidence for any differences in the genetics of female versus male ME, which is, I thought, interesting. But we haven't done the associations to the X chromosome, which may change our story on that.
Right. So that means, so the differences might be related to that, or they might be related to completely other things like hormonal factors or other factors. What would be?
Yeah, yeah, and we need to do much more complicated work to ask whether there are joint factors that perhaps one on a sex chromosome and one not on the sex chromosome that together work on changing people's risk. And we hope to do that, but maybe that'll be beyond the first paper.
So, with for patients hoping for some relief and treatments, I mean, I know it's impossible to say. I mean, any treatments that might arise out of this are still probably years down the line. Is that fair to say?
Um, Yes and no. The yes is that is the normal process for disease, and genetics doubles the chance that that process will be successful. The no comes from the fact that one of our collaborators, Precision Life, has already shown through their methods that there is a commonality to ME and long COVID genetics. And secondly, that they have taken what they've done for ME and are already starting clinical trials in the US. So if that pans out and turns out to work well, then they will have broken through and provide
A clinical trial based on
based on genetics
found in found in your study or found in
and found in their study of combinatorial genetics. And we've also given them DecodeME data for them to take that forward.
okay. So it's possible someone will do something and find a clue and decide there's some compound that actually can impact that process, and that may jumpstart uh, treatment possibility.
All of this is possible. Is it likely? We just have to wait to see.
Okay. Okay. Thank you, Chris.
Thank you.